my name's Laura. I'll be reviewing the topic of functional genomics for your final. So functional genomics is a genome-wide approach to study how the genome is related to an organism's phenotype. So to do this, we're going to make comparisons between entire genomes of separate populations. So this is male or female, cancer or non-cancerous patients. And we can look at how changes in the DNA and in RNA expression can alter genotypes through the following methods. So that's DNA and RNA microarrays, genomic resequencing with DNA, and RNA sequencing. So I'm going to go through the methods real quick. DNA microarrays are used to detect SNPs. So the first thing that we have to do for a microarray is fragment the DNA of choice into short single-stranded pieces, and we're going to hybridize it or bind it to pieces of DNA on the microchip, which are single-stranded known pieces of DNA. The SNP of interest will be the last hanging nucleotide of the fragment that's bound to the chip, and a fluorescent tag will be added to that SNP. So when we take a picture, the fluorescent tags will all show the complementary pair to the SNP of choice that we're looking at. You can also see if something is homozygous or heterozygous for that SNP by seeing if there's a mixture or a blend of fluorescent colors, because you're adding the entire genome. RNA microarrays are used to detect levels of gene expression throughout the genome. So you will start by creating cDNA from the RNA by reverse transcription of that RNA into DNA. You're going to add fluorescent tags to the cDNA pieces first. So for example, if you add a red tag for all of the cDNA from one cancer patient, and then you add green tags for all of the cDNA for a healthy patient. You would then mix these and hybridize both of the patient samples into a DNA chip with a single-stranded known DNA, and then you would image the chip. And you would see an image similar to here on the right, and the red indicates all of the RNA expressed by the cancer patient, the green by the healthy patient, anywhere in between is RNA expressed by both of them. So overall, microarrays only test for SNPs or the expression correlating to the known sequences of DNA. So for genomic resequencing, it's pretty straightforward. We'll start by doing an Illumina sequencing of the entire genome and compare it to the known genome sequence of that species. So the picture here you can see is just an example of a single strand comparison between a sequenced C. elegans DNA and known. And you can see the red indicates SNPs or areas where it doesn't match up. You can also see if you do multiple strands, if the person is heterozygous or homozygous as well for certain things. RNA sequencing is similar to genome resequencing, but we just need to create cDNA from the RNA first before we sequence it and align it to a known piece of DNA. So in red is the cDNA from the RNA that is matching to the known DNA sequence. So the red is an example of all the expressed RNA that you had in the cell. And if you have many copies of that cDNA from the RNA that's matching to the DNA, you know you have high transcription. If you don't have many copies, or if you have none, you know there's very low transcription of the RNA. So here's an experiment to think about these kind of techniques. I'll just read through it. MET-HB is a disorder of the blood, and it causes an excess of methemoglobin, which is hemoglobin with oxidized iron, which can lead to a shortness of breath. It can lead to hypoxia and can even lead to death. So Dr. Jules wants to see if there are any inheritable factors that can put people at risk for MET-HB. If MET-HB is a very rare disease, what would be the best way for her to pick patients to study? So we talked about this in lecture a little bit. For really rare diseases, it's easier to pick MET-HB patients within a family, because the chances are that if there are any inheritable aspects of MET-HB, it will be expressed more in a family. We would not use a GWAS or genome-wide association study because it would be very difficult to find people affected by MET-HB. Right, so Dr. Jules does not know if the cause of MET-HB is from SNP duplication, deletion, or expression changes. She doesn't know what it's from at all. What method should she choose to determine if there's any genetic basis for MET-HB in humans? So she may decide to use genomic resequencing or RNA sequencing if she's interested in more than getting the SNPs for this experiment. So she wouldn't do DNA microarray because she wants to know for sure if it's more than just an SNP. She does not want to risk using the RNA microarray here because she doesn't know if a microarray will have the corresponding single-stranded DNA in her target gene, and she wants to be able to tell if there's a large-scale change in the DNA, such as a duplication or deletion. So she will choose genomic resequencing and RNA sequencing, for example. So Dr. Jules does these experiments, and compared to a healthy genome, she finds that MedHB patients have a decreased mRNA expression coding for a reductase enzyme. So this is needed to reduce iron to its active form in hemoglobin and form RNA sequencing. And this was from RNA sequencing data. She also found a large deletion from what she thought was a known intron, and that was from Genomeresequencing. So what conclusions may she make about the cause of MedHB? So we know that this deleted sequence might have something to do with mRNA expression. So this mRNA, once gone, decreased expression, so it could have been an enhancer for that reductase enzyme. Also, Dr. Jules can't really prove for sure that this is the cause of MedHB, but she can definitely say it's correlated with the MedHB phenotype in the population. All right. Thank you all for listening to this review of functional genomics, and good luck on your exam. Thanks for listening.